Molecular drug imaging is a technique with which you use a drug and you couple the drug to a radioisotope. And that's sort of a flag that gives light. So you can inject the drug into the bloodstream of the patient and you can follow it on a PET camera. So you can actually see over time where the drug is going in the body. So you can see it whether it ends up in the liver, in the kidneys, but also whether it ends up in the brain. And that's something I think that's really important for especially also new drugs that are developed, whether actually these drugs enter the site where they should act, so in the brain tumor. And in Vuurden Medical Center in Amsterdam, we've just performed a study in DIPG patients in which we looked at bevacizumab, which is a drug, Avastin, used in children with DIPG, and we coupled it to zirconium, which is an isotope, and we saw that in some children the drug ended up in the tumor, but also in some children the drug was completely negative. So it didn't end up in the tumor, it just ended up in the heart, in the liver, but not where it should be. So I think this new technique of molecular drug imaging can help us doctors in the future to select patients based on this scan that might benefit from a therapy and avoid giving certain drugs to patients that do not benefit from a therapy, and thereby also avoiding unnecessary toxicity. I think it's been around for, I think, 5 years now. There is some experience in the adult population, women with breast cancer, for example. They've used this technique with trastuzumab, and there they saw that some patients have metastases which show up on the scan, so then you know certain metastases can respond, but also that there are metastases that are negative. So in these patients, maybe you should not give such a drug. And in effect, our hospital in Amsterdam also has done this study with erlotinib, which is another drug also used for DIPG, but they've used this in lung cancer patients. And using this drug and this imaging technique, they could see patients that had strong uptake of this drug and also patients that were completely negative. And these patients that had strong uptake, they reacted on the drug, whereas the negative patients did not show any effect. So I think this is really very important, not only for children with brain tumors, but also for children with other kinds of solid tumors, so that you can actually know whether a patient needs the drug or not. Well, the development of a radio-labeled drug is quite expensive. It's 60,000 euros to develop a radioisotope of a drug. But I think when you know beforehand which patients do benefit from a therapy from the patients that don't, I think it can also be very cost-saving. So in the end, a pharmaceutical company can know which patients do benefit. So I think in the end it will pay itself. So what you do is you give what we call microdose, so that's 1/100th of the actual therapeutic dose, and that microdose is coupled to the radioisotope, so that's what you do before you treat the patient. And if the patient has good uptake of that radioisotope in the tumor, then you can give the patient the therapy with the drug, which is the normal therapeutic dose. Well, I think molecular drug imaging is quite still in its early stages, and I think it needs international collaboration. It needs also infrastructure for a department, so you need a cyclotron, for example, to produce the radioisotope. But I think it's important that such meetings as we are now here, we can discuss with colleagues and try to set up these networks. So I think that's important for the future.